I'm very pleased to be here to introduce these three brilliant scientists. We are doing a workshop on biotech and medicine. We're going to talk about top breakthroughs in these fields and give you another perspective. We've already talked a little bit about some of the breakthroughs that you should expect over the next 3 years, 5 years, 10 years in both biotech and medicine. And today we have Stuart Kim, who's a professor of developmental biology and genetics at Stanford School of Medicine. We have David Haussler, who's a professor of biomolecular engineering at UC Santa Cruz, expertise in bioinformatics. And Michael Snyder, who was recently recruited to Stanford as the chair of genetics, and he's an expert on genomics and personalized medicine. So without further ado, I'm going to hand it over to Stuart, and he's going to get us started.
Thanks, everybody, for having us. Biology and medicine is right now undergoing this singularity phase shift, we think, and that's perfect for the kinds of things that you're talking about here. So give me an opportunity to get together with these two guys. So I've doubly enjoyed this because David is not only what you just heard, but he's king of all of the bioinformatics in the country, basically. And all of the DNA sequence flows into his computer center at UCSC. And so he's sort of watching all of the developments, and he has the best pulse on where we're at and what's going to become limiting and what it'll take to keep track with all of the information. So he'll tell you about that. And Mike's king of making all of the sequence data that David is storing at UCSC. And so the three of us have been getting together and talking about what is going to be possible tomorrow that's not possible today. And given that it's probably going to be possible in the 2 to 3 year time frame, how is that really going to change the way we do medicine or the way we think about ourselves or even broader implications? So that's what we want to talk to you about. Now the format is going to be like, one presenter and then 2 color commentators. And so it's going to be very interactive up here. And we want this to be interactive with you too. So if you have questions, if you have ideas, we want feedback. So we want this to be much like a town hall. I've given you what we're, at least a roadmap of where we might go. So you have the outline, and then you have the PowerPoint presentation slides, which is not necessarily the exact things we have to go, because if you have questions, we might be up talking on side issues. That would be great, okay? So without any more, what I want to do is I want to start by talking about the 2 technologies that are really just changing everything. One is the high throughput genomics that gives data on lots and lots of people and what that could mean. And the other is the computing needed in order to handle that data. So first Mike will tell you about the hardware, and then David will tell you about the software changes that are occurring. Okay?
Okay, so as Stuart said, I'm going to review a little bit about DNA sequencing. The first slide will be very, very basic to make sure we're on the same page. And also, as Stuart said, please feel free to ask lots of questions. So I think you've already learned about DNA, but again, this is by way of review. We all have similar but slightly different DNA, except for identical twins, which are nearly identical in their DNA. And just to remind you, we have 2 copies of each of our DNA of 3 billion bases, and it's distributed on 23 chromosomes. So we have 2 copies of each of 23 chromosomes. And together, this is called a genome. That's our genome, 6 billion bases. And so when you hear somebody talking about sequencing a genome, it's determining the sequence of all 6 billion bases. This was first done, and a rough draft of this was completed in 2001, and then a more complete form was completed in 2003. This was done from a collection of individuals so that there's not actually one person's genome sequence that was initially done. It was done by an amalgam of a fair number of genomes, and they call that the reference genome. And so when you get your genome sequence, it's usually done relative to this reference genome. Now, when the reference genome was first sequenced, there was a lot of work. It was 2,000 people involved. They slugged it through and ultimately were able to complete this at a cost that's somewhat difficult to estimate, but somewhere on the order of $0.5 to $1 billion. And it used machines that would process 384 samples at once, and it would determine, you probably heard your DNA is made of these, I said 3 billion bases, it would do 1,000 at a time. Okay, and you had to slug this through and do it many times over. What's happened in the last few years is there's been a revolution in DNA sequencing, in part inspired by the National Institutes of Health, which is trying to get to sequence genomes for $1,000. So get your DNA sequenced for $1,000. You may have heard of this. And we're not there yet, but there's been an amazing amount of progress in the last few years, which you'll see in a minute. So some of these new technologies, in fact, virtually all of them, process millions of samples at once. Although the amounts of DNA determined in any one so-called read is on the order of about 30 bases and can run up to maybe 500 bases depending on the technology. But the key is that even though they're fairly short, so you're determining, say, 30 bases at a time, you still are able to, you determine millions of these things at once, and together you can deduce a genome sequence. We won't get into the details of this, but these are the technologies that have come along in roughly the order that they've come out. So what's happened, and I think you've probably learned this already, is that the cost of sequencing has really plummeted. And this is, in fact, much, much greater than Moore's law. Probably all of you know about Moore's law, with doubling every 18 months. What's happened starting around 2004, you're getting about tenfold every 18 months or so in technology. And if you look at what's coming along, it's not going to stop for a while. So the latest, most of these companies, by the way, are in the Bay Area. And the latest one from Complete Genomics, we can get a genome sequence for $20,000. Okay, that's the current going rate, and if you bundle a bunch together, it's even less than that. And they claim they can do it for about $4,000. So with this kind of cost and throughput, they figure they're going to actually sequence 10,000 genomes next year. So this is the throughput that's coming out. I forgot to say, so with this drop in technology, this is the number of what are called personal genomes that have been sequenced. So here's the first one that came along in about 2004 or so, then a second one, this is cumulative, and then so on and so forth. This actually has become out of date. 3 more just got released this past week. So we're up to 11, and as I say, imagine next year from 11 to 10,000. You can see the progress we're talking about here. Okay, so many of us expect that, in fact, in a fairly short period of time, there'll be 1 billion human genome sequences out there. And probably everyone in this room will have the opportunity to get their genome sequenced if they so desire. And that's not 1 billion people, but imagine that's less than 1,000. So imagine that many, many times over. Okay, I have to say what's going to be rate limiting on this, David will tell you, the computation, it's not going to be the sequencing that's rate limiting. A lot of it's going to be the computing. And quite frankly, the biggest rate limiting step is going to be having good data that goes with each of these sequences, what's called phenotypic data. So medical histories and things like that, so that you can really use that data. That will become the most limiting step in this whole process. So anyone who does that well will in fact have a leadership position. This is just a few examples of people who had their genome sequenced. This guy was first, Watson was second, and here's a local, Steve Quake, just had his release this year. Okay, so again, these sequencing machines, they generate lots and lots of data. We need other kinds of data to go with it. Yeah.
What's the error rate on those sequences that get to 11,000?
Yeah, great question. It varies with the technology, as you might imagine. So 454 is running somewhere around the 1% error rate. And it has a very distinct kind of error. It drops bases or adds extra bases where there's tracks that are exactly the same of bases. Other ones like Illumina and some of the others, theirs is, it's good in the first number of bases, but as you get further and further away, then the error rate starts skyrocketing. So it depends where you are. An average error rate would be somewhere 1% to 2% for the lengths most people use. And the key to this then is that they usually do 40-fold extra coverage so that they can, if there are errors, they can basically get the same sequence many, many times and try and correct for that.
So our Archon X Prize for Genomics has a 1 in 100,000 error rate. When do you think that will get hit?
That's a good question. I don't, some of these companies are claiming they're close to that now. I don't know that that's true, but there's another big issue you may be aware of. This is just single base errors. The thing that's hardest to figure out in a human genome sequence aren't the single bases. They're called SNPs, or single nucleotide polymorphisms. I don't know if you've heard of those. But it turns out the most challenging part of determining a genome sequence are something called structural variants, which are chunks of DNA that are deleted or duplicated or flipped around. And actually, there's more of that going on in the human genome than single nucleotide bases in terms of the amount of bases that are affected. So the structural variation stuff is very, very hard to actually decipher. And working that out isn't close. So when people talk about, in fact, I talked about these human genomes getting sequenced, that's usually for the single nucleotide polymorphism. These structural variants are actually very poorly deduced, if at all, in some of those sequences. So they're not even complete sequences.
And the other challenge, of course, is validating it.
Yeah, validating in a sense showing accuracy, yeah. My own view is at the end of the day, it'll be a combination of technologies at its best, because each of these different technologies has different kinds of errors. And so if you actually put them together, your error rate should plummet quite a bit, except the structural variants are still very hard to get.
What is the time frame right now, and do you think it'll be a year from now to actually sequence one genome? So if I submitted my DNA on Monday, will I have my DNA by Wednesday?
Not yet. I think we can get all your reads done probably in about 10 days, meaning all the actual sequences. But then somebody's got to put it together and analyze it. And that's going to take at least a month. That's a minimum backlog. But to look at it carefully, it'll probably be a few months. With some of the technologies coming, there's one, for example, PacBio publicly says that they anticipate sequencing a genome in 15 minutes. Okay, so that'll certainly do the sequencing. Then it's going to be up to guys like David, again, to help you interpret that sequence and put it all together and then interpret it. So the sequencing won't be rate limiting. It'll be the next steps, finding all those things that will keep you up at night. And David will tell you about some of the challenges associated with the computational side of this.
All right, thanks, Mike. So this is the obligatory slide that we've already gone over. We'll just skip through it, but it shows that same kink that we saw where the cost per base in DNA sequencing is actually falling faster now. We at Santa Cruz actually participated in the first draft of the Human Genome Project. We put the first draft of the public version of the human genome on the web in July 7th. This shows internet traffic outside. This is the total internet traffic leaving the UCSC campus, and that's the moment we put the first draft on the web. We are far exceeding the needs. Right now, our main problem is getting data down from the NCBI, the central repository for genomics data. We're trying to download on a daily basis gigabytes of data that indicate tumor genomes, which we'll talk about in a while, which are in addition to personal genomes, personal germline genomes, something very important. And they have tools like Aspera and so forth that try to get you enough information over the web. But the web just simply was not designed to transfer this kind of data flow around. And so you really have an immediate problem with step one, just moving the data, let alone analyzing it. So we will need a formulation in which the data is distributed and analyzed in a distributed way, and the cloud, for those of you familiar with cloud computing, I'm not going to give an intro to that, but cloud computing is certainly a possibility there. Just again to illustrate the change in computing size, this is actually a picture of the little rack of 100 Dells where we assembled the first draft of the human genome based on the information that we got from the 20 genome centers in 2000. And it's funny to look back on that, little piece of computing equipment. The kind of scale that we need is better, that we really need to understand these data is better iconified here down below. This is the Google data center where you have to locate it next to a river where there's cheap power and so forth. It makes a lot of sense, but really, in addition to this level of raw power, the algorithms to really understand what's going on in the computing, in the process of computing information from a genome will need extraordinary development over the next couple of decades. So we will see this a little bit, I think, when we get to cancer, and I'm going to return to some of the computational problems of interpreting genomes then. But we want to move along very quickly here. So I'm going to hand this back. Are there any questions?
Is there a way that Google handles internet data differently than the way you handle DNA sequence data? I mean, is there anything fundamentally that needs to change at UCSC from the way that this DNA storage, the DNA is stored or interrogated or hooked up together?
One challenge is just indexing everything in a creative way. Google has managed to do that with text so that you can, any reasonable person can type in some keywords and get to the information that you want. But indexing all of the genomic data in the world is a different story. It is like a text, but it shares evolution. So you really have to index it based on how it evolved from common ancestral DNA. And it shares, in the particular pieces of DNA, share clinical information and other scientific information that are associated with them. And so you want to index it in multiple different ways. So it is a fascinating indexing challenge.
One of the things that I heard about recently was that the cost of generating a gigabyte or a terabyte of genomic information is actually dropping incredibly quickly.
Yes.
But of course, hard drives still have a cost that currently is less per gigabyte, but might not be
Yes.
there forever. How do you intend to be able to grow your data architecture to keep up with this new information?
It's a very challenging problem to build this index that would keep millions or billions of genomes in a way. One of the things you want to do is exploit the fact that we're all related to each other, and Stuart will be talking a little bit more of that in terms of common ancestry. And so you actually don't have to store the same snippet of DNA again and again and again. You can store the differences as it's passed down, and that's a huge compression. But there's actually some very interesting and I think it sounds whimsical, but it's very serious. The best way to store DNA is as DNA. And so if you think about storing all these genomes that we're sequencing now and then how good the technology is going to be 100 years later, or 10 years from now, sorry, not 100, but 10 years from now, I can imagine thinking, wow, I want to retrieve this genome. Well, should I pull up a terabyte of data from this half-assed old sequencing machine? No, just give me the DNA and I'll use a modern machine and I'll get the genome sequencing much easier. I guarantee you'd rather have the DNA than whatever we're sequencing today 10 years from now.
You don't actually store the methylation states.
So yeah, methylation is part of the DNA that when in most storage that's a covalent modification, so it would be available in the stored DNA as well. And new sequencing technologies will probably have the capability of reading methylation. I can imagine that's going to be the thing. We have indirect ways of reading it now.
Thank you.
Yes.
A slightly more ignorant question, sorry. You do sequence and store what is popularly called junk DNA as well. Is that
Yes.
right? Thank you.
Absolutely. And my lab in particular believes there's a lot of gems in the junk. We do a lot of work on that. Yes.
It's not clear what's junk.
Yeah, it's really not clear what's junk.
Yeah, you mentioned that you're downloading gigabytes per day from NCBI. Is that compressed so that you're only getting the deltas relative to the
No, see, we haven't worked out a decent scheme so that we can get compression relative, even simple compression relative to a reference would be a lot easier. But we're downloading raw reads because we want to dig into the data deeply, and we don't believe all of the assembled kind of final information. And that's what really amps it up. So we're downloading data that covers every base up to 100 times. Yes.
And what about the privacy aspect of storing that DNA?
Also a huge question. Yes. Yeah. I mean, the people are touchy about privacy, and for many good reasons. So one has to be careful, yes.
Mike, do you have any information about the 1,000 Genomes project or anything like that?
In terms of privacy?
Yeah?
Those lines have been around for a long time, and people sort of think it's okay to do this. I mean, this is a big issue. It might actually be paid to talk about this when we get to the ancestry and things like that, because you'll get some comprehension about how close people are to each other. But don't let us forget that point. It's a great question.
It's really interesting on this privacy issue because there are some people that guard their DNA sequence like it was, they guard it. And there are other people that publish it on the internet, like those three people that Mike showed, like Steve Quake. I can download Steve Quake's genome sequence. There's 173 deletions in Steve Quake's genome, and he's just proud to show it to me. On 23andMe, which I'll show you in the next slide, people are trading genome sequences like mad. I get, it's like Facebook. People I don't even know are saying, let's be genetic friends. And they're just piling up. They're piling up on my inbox right now of people who want to be my genetic friends I never heard of. And people are just trading genomes to see, on the off chance that we're how related we are or what parts of the country we come from. So.
Yeah, I wasn't going to get into that. But, even Jim Watson, who put his sequence up there, didn't put up his ApoE, which is implicated in Alzheimer's. So even he held a little bit back. So you can see that, people will be happy to put a lot of stuff up, but they may not want to put 100% up. So you may want to look at your sequence first before you put it up. It's possible. You have all options, but a lot of people, as Stuart said, it's easy for academics because we have tenure in healthcare and such forever, so we can put our stuff up. It's not a big deal, but it is a real concern. I guess if you really want to get into this, you can't be discriminated on employment or in terms of health now based on your DNA sequence. But you can still be discriminated on life insurance. So technically you could lose your life insurance if you had the wrong thing.
And you're making a decision for your children because they're getting half of it. And so by making your genome public, you are making half of their genome public.
You've said
brothers and sisters.
Yeah.
You said the best way to store DNA is as DNA. Are there any techniques being developed to effectively data mine DNA in vitro, comparing it to DNA, rather than trying to convert it over to digital formats? Is there work being done to try and effectively do something we do digitally, but do that in a biological kind of way? Like data mine, compare DNA against each other?
Well, a lot of the technologies work on hybridization. When people read DNA by hybridization, by matching using base complementarity to read DNA. So that is a direct way, and there are other biological manipulations that do that. Again, it's we're building machines that interface. The PacBio machine just reads a strand of DNA, basically. A single strand, right? And so that's the kind of machine you want that just kind of interacts very directly with the DNA.
Okay, so I'm going to introduce the next subject, which is all on personalized medicine. So this is my file from 23andMe. And if you click one page down from the main page, you get to this thing. So up here it says, it's my name. And here it just tells you disease risks, carrier status, traits, and drug response. So what we're going to do is talk about these sorts of things and specific things that are about to happen in the next 4 or 5 different sessions. So, I don't have any huge disease risks. This thing on prostate cancer, it's a small risk. It's a little bit bigger than a small risk. I don't have, I'm not a carrier for BRCA1, so if I have a son, but even if I didn't have a son, if I had a daughter, it's not getting BRCA1 for me. Not a carrier for cystic fibrosis, so that came pretty good. Traits, I found all sorts of traits that I already knew about. So I knew that my eyes are brown and that that I flush when I drink alcohol. I can tell that real fast. So we'll be going through some of these things in a minute. Now, it's really interesting to watch personalized medicine right now because as far as I can tell, there are 3 things. There are people who may or may not want to get their genotype. There's personalized medicine, and there's the med schools, and whether or not they can use that information to help you. And there's the scientists who need a certain people genotyped with their phenotype in order to figure out how it all works. And they're all interconnected. So I'll explain why. Scientists right now are having trouble figuring out which genes are associated with which things, because it's hard to get enough people. So getting 30,000, 50,000, 100,000 people genotyped in one experiment is pretty expensive. And so the scientists are having trouble getting enough good information on how it works. Because the scientists can't figure that much out, we're making some progress, but the big discoveries are yet to come. The doctors don't have that much to talk about. So the doctors are reading the literature and they're saying, well, how much is this going to affect my patients? Now, there are some clear big winners here, like warfarin is a really big winner. BRCA1 is a really big winner. But like I said, the potential is enormous, and we're just beginning to see the whole potential. And because the scientists haven't figured out the science, and the doctors don't need to use personalized medicine when they treat you, there's no reason for a person, there's not that much reason for a person to get tested. Now, if that logjam gets broken and any one of those three things happens, it can all happen. And that's where we're all optimists, and we think that a significant fraction of all of the first world will be genotyped because somebody's going to figure out the science, or we're going to start teaching personalized medicine at Stanford, or 23andMe. All we need is one break in the logjam. So if somebody figures out a killer app at one of these personal genomics companies that can get 50 million customers, and then they can let the scientists use that to figure out the science, it'll all roll out. So one killer app is what we're away from in terms of this stuff really working big time. So 23andMe is going with Ancestry, I would say, and that this is where you see a lot of the activity on 23andMe, and Navigenics is more on health. And these guys are trying to figure out what is the killer app I can sell this stuff with. Okay? So David will now talk about preimplantation and prenatal genomic analysis.
Okay, so talk about diving into the controversial stuff. So if you accept the fact that we will be able to sequence genomes and we will understand a little bit more than what we do now, what are the possible applications? So what are the society-changing things that we might do with genomic information? And I think the first thing, which has enormous social implications, we have to think about are diagnostics that relate to birth decisions. In particular, you have several avenues now where you can actually get tested. It is an invasive procedure. If you think about the in vitro fertilization clinic procedure, right now, if you are at significant risk for carrying and passing on a devastating genetic disease to your child, it is actually becoming increasingly common to go the route of in vitro fertilization. You will stimulate the mother with hormones. She will ovulate, superovulate. Each of those will be artificially inseminated with sperm from the father. It will grow to this stage that you see here as a tiny embryo consisting of just a few cells. And then you can take one cell out with a pipette, as you're seeing here, and do PCR to test for a few hundred different conditions. And if the embryo tests well, it doesn't have this devastating genetic problem, then that gets implanted back. The fact that it lost one of 8 or 10 cells is not going to affect the development of the child. So this is a scheme for how these pre-implantation diagnostics work. And what will change radically in this field is that instead of being able to test for a few hundred specific mutations, which are the approved tests at this point, and normally only 1 or 2 is applied in any particular case, it is possible with technology that could be developed, but doesn't exist today, from a single cell to possibly do a whole genome scan. And the implications of that are something that would be very interesting to talk about. As Stuart pointed out, we would be hard-pressed to interpret that information at this point, very hard-pressed. And so one would be loath to make life-altering decisions based on a completely half-baked interpretation of those data. Probably more appealing than this in a large setting, a large public adoption, would be non-invasive tests. And so you might not be aware of this, but in fact, while the fetus is growing, and even at early stages, the fetus sheds cells which then appear in the mother's blood. They are very, very rare. 1 cell per milliliter of blood is a tiny, tiny, tiny fraction. So we're talking like 1 out of a billion type odds of finding one of these cells. They are largely nucleated blood cells, which are highly transient. There are actually interesting types of cells that can persist for years. Actually, white blood cells can show up years later in the mother that are actually fetal derived and cause some interesting immune issues. This completely, so this is absolutely would be the Cadillac of diagnostic procedures. It's completely non-invasive, and if we could do single cell genome sequencing, you would know everything there is genetically to know about the embryo. And if you can do this at 5, 7, 9 weeks, this is very, very early in a pregnancy where many women would be more comfortable in making a decision whether to keep the child. I think this has enormous social implications, but again, this is a futuristic technology. We can now do PCR and test for a few different things from those cells, but it's very hard to even get them in the first place. What's actually coming into market, and there are companies based on this, is cell-free DNA in the mother's blood. So this is DNA that exists outside of cells. It's just floating around in the blood. And I was surprised to learn in researching this that it actually constitutes 3% of the mother's circulating cell-free DNA in the early stages of pregnancy. It consists of short pieces, less than 200 base pair, whereas this is the fetal-derived cell-free DNA, whereas the mother's own cell-free DNA is in longer pieces. So if you do a gel-based DNA size selection, you can greatly enrich for fetally derived cell-free DNA, more than 50% of your DNA. And then you can sequence this and diagnose something about, again, the embryo. And Steve Quake's company, who you saw earlier, has posted his genome, has got a method to diagnose Down syndromes. Down syndrome is an extra copy of chromosome 21. So if you get too many reads, fetal reads from chromosome 21 relative to the other chromosomes, then you know this child has an extra copy and they will have Down syndrome. There are other things that you can test for, but mostly you can test for paternally derived mutations. Otherwise, you're very confused between the mother's blood and the fetus's blood. So if there's something distinct about the father, you can test for it by PCR. And then you get, interestingly enough, there is a phenomenon of the vanishing twin. Usually these other embryos are lost quite early in the pregnancy, but they produce fetal cells during this critical time, and you get 2 signals, which can be very confusing to these methods.
Can I ask a quick question?
Yes, go ahead.
So
just winding up.
When you're looking at the fetal DNA, that assumes that's all you have to work with. But what if you had both parents' genomes fully sequenced? I'm curious. Obviously, that wouldn't work to assess whether the child has Down syndrome, because that's just going to be an extra copy of the chromosome. What percentage of the genetic tests that you might want to do on your child could be just determined based on the possibility, based on carrier status, that sort of thing?
Very good point. Absolutely. So we are in a position very soon, within a few years, of having the possibility of sequencing both the parents before you do these tests, and that will focus you in on what you want to look for. So they will focus in on specific combinations of alleles that you know could have been inherited from the parents and would cause trouble. And that will be a very, very important thing, and I think that will be an absolute requirement for really getting a lot of the information about this. But you still need to sequence the fetal DNA if you really want to know what combinations are actually present in the embryo, because you have a 50-50 chance of each getting one of the two copies at every locus. Now they're linked, so you can infer kind of what you got nearby from certain marks. But if you're looking through the genome, you really need to know which copies you got from mom and which from dad. Yes.
What kind of verification are you using when you find like the quakes work on a Down syndrome, and you come to a patient and you say, their risk is really high. Have you done enough verification work?
Yeah, absolutely. You want to know exactly what is the accuracy of the test.
But how do you do the verification? I mean, what
So
was your
they
process
have
of
blind
verification?
tests. So if you read Quake's paper on this, they did a bunch of blind tests where, you don't, the method wasn't aware of whether it was a Downs or not, and then there was careful validation to learn what the truth was, and it has a certain accuracy, and I can't remember how highly accurate it is. In the 90s somewhere, I believe. But there is, obviously
there
you
was
have to
good
very carefully
statistics on it.
quantify the accuracy of these tests.
See, I think the idea is the amniocentesis test, which is possibly more accurate, is also dangerous for the baby.
Right.
And
Right.
so, you could know that the baby was normal, but you have a chance that you're going to hurt the baby. So the idea is just to take a little bit of blood, sequence the blood, and then the baby is fine no matter what, and you still have a pretty accurate test.
Okay, thanks.
Thanks, Stuart. Yes.
Couple things, having just lived through that. I'm 8 months pregnant. It's 3 blood tests. They do 3 blood tests first trimester, so they hit it, and I think it's above 90%. Amnio is 100%.
You had a Steve Quake sequencing?
No, well, they've got, I didn't. That
different
was the
ones.
problem. I was 17 weeks by the time I got in there, so I had an amnio.
Uh-huh.
But that's what they're saying is there's a series of 3 blood tests that they can do early on in the first, kind of in the first trimester.
Yeah.
And come up with like a 90 or 95% reading on Downs. And then the amnio is going to give you 100%, but there is the risk of miscarriage.
Based on a blood test, yes.
Right.
Yeah, the Quake technology is not the only technology for doing Downs analysis from blood.
But then secondary, and actually I wanted to address the logjam point that you brought up. The one thing that you didn't mention that I think is worth noting is the pressure that the FDA is putting on drug companies now, and the whole difference between clinical medicine and clinical research. When you look at these companies, especially the companies that are looking at blockbuster drugs, and they're running it through thousands and thousands of patients, there is, because of the nature of research, always the chance that you're going to because of the blinding, you're going to misclassify your patients into, say, a high treatment group when they would be a low treatment group, or more importantly, a patient who would need a high dose would be classified into a lower dose and therefore not show up as having been efficacious. And that, I think, could potentially be a real pushing point to break the logjam if you can identify your patients before you classify them, thereby giving the FDA a reason to say, here, listen, we can do this, and then we
absolutely.
can specifically treat
Absolutely.
the people who are going to respond to our drug.
I think that's called pharmacogenomics, where you know something about your DNA, and that tells you something about how you respond to drugs. So warfarin is right now one of the best examples of that. So warfarin's a blood thinner, and it's dangerous both ways. If you don't have enough, you don't thin your blood, and then if you have too much, you can get bleeding and get a stroke. Right. And so you have to tune the amount of warfarin to the right amount for every person. But the gene that encodes the warfarin receptor is variable. And so different people respond to different levels of warfarin. So they've started to figure out which DNA variant of that warfarin gene encodes for the high-risk drug and low-dose drug. And so I happen to have a sensitive one, so I'm at increased sensitivity for warfarin. And that would be useful to me if I had to go in and be under warfarin treatment.
I literally was. I looked at my 23andMe and told the doc, hey, I'm in the sensitive group, and set the dose. I had a brief stint on it. So, that's one thing that really happened. Also, I think your question had something to do with clinical trials as well. And I think this is, I'll hit that in cancer, but one of the really first applications is picking the patients that will be most powerful in demonstrating efficacy of a new drug. And so these play a very strong role in clinical trial structure. Okay.
Next up is Mike.
Mike.
Okay, so just to lump this into two sort of segments of what you might do with a genome sequence. One is, and how it relates to personal medicine, one is predisposition for diseases, and David touched on that, and I'll touch on that here as well, but this also translates into the next part, which is monitoring diseases that have already happened. So one is being predisposed for disease, again, and having appropriate tests for that, and the other is dealing with the disease that's already formed. There are certain diseases out there that everyone recognizes, like sickle cell anemia or cystic fibrosis, that is very easy to identify, and you can follow it through its family history. But for a lot of genetic diseases, it's very, very difficult to follow. It's basically what Stuart was saying before. Most diseases that are out there are actually complex. So diabetes, obesity, schizophrenia, and even simple traits like height are very complex. There's no one single gene that lets you tell what's going on. So do you know the best way to tell height? It's clearly height is genetic. What's the best way to tell height? Look at the parents, right? Same is true, well, diabetes, you can actually just follow glucose and such. But there have been studies, for example, in diabetes where they looked at 30,000 diabetics and 30,000 normal people, and they map out all the genetic contribution. They mapped out 18 regions of our DNA that are responsible for this, and they found 7% of the genetic contribution. So genetics can tell you something, but it's hard to actually figure out, probably because there are what are called rare alleles. There are rare cases where these things, and they probably combine in certain fashions to give this. Or alternatively, it's environmental gene interactions that are responsible. So there clearly need to be other ways of using this sorts of information. And some of the ways that are coming down the road that we like to think is happening, this has not yet been shown, is to combine genetic information with other sorts of information. So you've learned about proteins, you've learned about RNA yesterday, but there are ways of following the expression of all your genes by following RNA levels. There's something called DNA microarrays you may have heard of, and there's a new technology sequencing base called RNA sequencing that lets you measure the expression of all of your genes, okay, in any particular tissue, like in your blood or in the affected tissue. And likewise, there are new technologies people are working very, very hard on to follow protein levels for all the proteins you have. And there are 25,000 or so genes, so there are 25,000. There are actually more RNAs per gene. Not each gene can make multiple RNAs. And likewise, you can get many proteins from every RNA. So there are now technologies to follow thousands of proteins. And likewise, there are technologies for trying to follow metabolites, like the sugars and lipids and things like that that are in your blood. And so currently, if you get a blood test, they typically test, well, there are some doctors here, you probably know better than I do, they typically test somewhere from 10 to 30 different things in your blood. That's pitiful if you think about it, because we have thousands of things in the blood we should be measuring, but we're only measuring 10 to 30 things. So what's coming down the road is that people like the idea that you'll be measuring these tens of thousands of things so that you'll be able to see signatures from this that basically tell you what's going on with your so-called disease states. You can follow RNA levels for thousands of RNAs. This is already being done. You can follow protein levels, and the technology is called mass spectrometry, which again lets you follow thousands of these things at once. And specifically, people are looking for what are called biomarkers, particular proteins that will be reflective of a disease state. But most of us like the idea that it's not going to be one protein that determines it, that will give you a perfect signature or perfect diagnosis. It'll be a combination of markers. And actually, I'm involved in a company, so I do have a conflict here, that's making antibodies to all human proteins, so you can follow all those at once. It's a way of measuring levels. And as I say, metabolites, should let you measure the metabolites for lots of things. This would be done by mass spectrometry as well. So the thought is that what you'll be able to do is combine genomic information, and you add in information about proteins, the levels of thousands of proteins in sera or in the affected tissues. People like to work with sera, urine, although maybe in the future, breath would be a way to go to look for metabolites and proteins. And use signatures that come from these states. So asthma, for example, if you think about asthma, breath would be a good thing to use. And then from this, so maybe you only get a 3% predictive value from this, but you start combining information from thousands of levels of thousands of proteins, and then you try and predict a so-called normal person from maybe a disease state, as is shown here.
That's a normal person?
That's your typical person.
Normal person of the future.
Right. There you go. So anyway, that's the concept. There are many, many people working. So there are a lot of people working on these single markers, but many of us like the idea, again, you'll be following thousands of these. And what's also equally remarkable, you may have heard this idea already in this, is that a lot of these technologies, the machines we use now are really quite big. But a mass spectrometer is the thing they use in an airport, right, when they're looking for bombs. Those aren't that big. The ones that they'll take, they do a wipe and they walk over to the machine. The machines they use are fairly small, and you can actually make them handheld. So if you think about where the future is going in this, you could envision an iPhone type version to measure your, thousands of proteins or metabolites in your blood. You could prick your blood and ultimately get fairly accurate diagnostics. This is an area that I think many people would like to see this go, so you can figure out your disease state. If you're feeling a little depressed, you prick your blood, see your proteins and say, oh yes, I've got a so-and-so infection, or maybe this is happening today or something. So that's the pie in the sky view on all of this, but again, this is a direction the whole field is pushing towards. So, comments and questions?
You said that you could actually, you said you could measure the level of expression of proteins by the level of the mRNA in the cells.
Not exactly.
Indirectly. How accurate is that?
You can measure the level, they're actually separate tests. You measure the expression of the RNAs and you measure expression of the proteins. And the correlation, depending who does it and how it's done, is anywhere from 0.4 to 0.7. So there's some correlation, but it's not perfect. And that's because there are additional controls on RNAs to see what happens to them to get them turned into proteins. So it's not a perfect correlation, but it's some. Most people like the idea. There's been a big push to actually follow RNAs, but most people like the idea of following proteins because it's a much more accurate reflection of the state of a cell.
Now, RNA can be turned into DNA with one enzyme. And so if you just take any RNA and turn it into DNA, then your ability to look at RNA goes up exponentially along with every sequencing technology. So the RNA characterization is increasing exponentially right now too, in parallel with the DNA. I don't think the protein is changing quite so fast. I mean, that's progressing more linearly, wouldn't you say, with a mass spec? But the big jumps are coming in RNA and DNA characterization.
Well, it's changing quite a bit now with these new methods in mass spectrometry. You can definitely follow thousands of proteins at once now with some of these new machines, which in fact we've just bought. So,
thank you. I'll talk a little bit about cancer genomics now. So this is another application, and we want to distinguish between the kind of application where you come in and sequence your genome or sequence your child's genome once, and then you try to conclude from that, well, these are the cards they were dealt. These are the genes that they inherited. What does that mean for their health throughout their lifetime? That's one model for thinking about how to use genomics. But actually, there are diseases that arise from genomic changes that occur after you're born. Cancer is one of them. This is DNA damage in the cells that occurs later, and you have to keep sequencing. Many times you have to sequence different tumors. If it comes back, you sequence again, and so forth. So this is a completely different way of thinking about this, where you would do lots of sequencing from the same individual. We are just starting now to understand cancer as a genetic disease by looking at complete genomes. It has been long understood to be a genetic disease, and it's been studied by sequencing specific genes that were known to be important in cancer. But now we're starting to see the first data that show overall structure of the genomes inside the cells of the tumor. And each one of these green lines represents a piece of DNA that is connected abnormally. So we have pieces of DNA in chromosome 8 that are connected to pieces of DNA in chromosome 14. And so the DNA, the chromosomes have actually been broken up and stitched back together within this cancer in a process called rearrangement. sometimes that can actually create a novel gene. And so this is a novel fusion gene that's associated with an actual cancer outcome in this patient. This is a gene, part of a gene from chromosome 2 fused to a gene from chromosome 12. So this occurs, you can look at various lung cancers and prostate cancers. One of the most famous is the ABL-BCR fusion in leukemia. So there are many actual documented repeated fusions. The same thing happens again and is very important in driving the cancer. We will discover hundreds of new fusions that occur in cancer by looking at the genomes. But the genomes contain millions of other points of data. There are just copying a region and having an extra copy of it can alter the way the cell functions and make you susceptible to going the metastatic route. In tiny little point mutations all throughout the genome can also drive the cancer. So we are just now getting the first glimpse of this, and we are going to start thinking of cancer as what it is. It's not just something that happens to you once and then you treat it and it's gone. As many of you know, and I'm sure many of you have family members that have battled with cancer, it has this very bad property of emerging some kind of resistance. So you can be fine and you can be in remission and then it can come back. What's going on here is very analogous to what happens in when you have bacterial infections that come back after they become resistant to antibiotics and come back. It's actually an evolutionary process. There are billions or trillions of cells in the body. When a tumor forms, it creates an enormous number of copies of these cells, and the drug therapy or the chemotherapy might knock out most of them, but if there are a few that have specific mutations that make them resistant, then they will regrow a new tumor with resistant mutations, and that is very bad. Now, the good side of the genetics that result from this kind of evolutionary process of taking mutations and then copying them like crazy, and the way genes work within the cell is that often cancers acquire their ability to grow without bounds, which is definitely not normal for a cell, and other bad attributes by becoming essentially addicted to certain genes. So they are totally dependent on particular mutations or particular rearrangements, amplifications of genes. And so if we can understand from a genetic point of view what they're addicted to, what special thing they need to do their cancer thing that normal cells don't, then we can treat them by denying that pathway or that activity. In other words, all of these changes within the cancer genome create genetic signatures that hopefully will suggest that they will be sensitive to particular drugs or combinations of drugs. And from an evolutionary point of view, and Mike will say more about this, the drug cocktail is the way to go. It's very hard. You can imagine evolving resistance to one drug, but give it 3 drugs that hit all different genes. It's very hard to evolve resistance to a cocktail. I'm going to give you a real quick view of some of the informatics that goes on. That says computational cancer genomics up there. This is informatics that we're actually doing at UCSC. So you can actually plot. What you're seeing here is each row is a particular tumor from a different individual. These are breast tumors. These are brain tumors. And you're seeing plotted here in color whether that region of DNA appears more than the normal amount in red or less than the normal amount in blue. So it may have been deleted. And you see, first of all, that even though each of these tumors evolved independently in different person's body, they tend to amplify or delete the same regions. In other words, similar genomic lesions are seen again and again arising independently and leading to a particular type of cancer, and each cancer is distinct. It has a signature of genetic lesions. So we are learning to classify and subclassify and sub-subclassify cancers by looking at these. And just for contrast, these yellow individuals are normal, and you can see that there's very little disruption of the overall chromatin copy number there. If you in this informatics platform, you can see all of these data. You can look at clinical attributes, not to get into the clinical data here because we want to be careful about patient privacy and so forth. I'm just showing you the age of the patient, younger here versus older. Already you see, though, there are different patterns that are characteristic of the changes that happen in the younger brain tumor patients than the older. After a t-test and Bonferroni correction, you can actually see that down below. And so what's interesting about this from the informatics point of view is there are hundreds, thousands of questions we could be answering about these samples. And you can, in the UCSC Cancer Genome Browser, you can formulate a question, divide your data into 2 groups, run a t-test, get an answer right away. So this allows a very quick loop of hypothesis generation and testing in terms of cancer analysis. We want to organize the data not as positions along the genome, but according to pathways. There are known pathways at which cancer genes interact with each other. We can organize it that way. We can throw in tumor mutations. We see that there are abnormal number of mutations in the tumors in P53. That's well known to be a very important driver of cancer. It's almost essential that you inactivate P53 or something else in its pathway before you can become a successful quote-unquote tumor. And most of these patients did show that mutation. There are other mutations which I won't go into. I think we're going to hear a little bit about EGFR, one of your examples, Mike. This is one of the pathways, the PI3 kinase pathway, and you can actually plot among the patients. They tended to have amplification of these genes in red, deletion of these genes in blue. And the interesting thing about this is that the logic of this, if you follow the pathway, this little T means inhibits, and this little arrow means activates. The logic of this is that all of these mutations tend to increase proliferation of cells or survival of cells. So the cancer is protecting itself. It's altering the genome to make itself grow. And all of these are within that. And you might not see the same exact change in each tumor, but you see a change to this pathway that has the same net effect. We're able to do that now. And this is a kind of a model for the clinical trials question. So one of the most important things now is to pick patients who will respond to the new therapy. And so then your trial actually demonstrates that that therapy works. And so you will see this kind of personalized cancer come out first in picking for clinical trials, probably before it gets to the general practice. And there are things like Stand Up to Cancer. Sorry, this is off the top, but this is you may have seen the STARS came out for recruiting money for cancer. We're involved in this with a number of other researchers, including Dennis Lehman, who's the discoverer of the first, developer of the first real targeted cancer therapy for breast cancer, Herceptin, targeted against HER2 positive patients. And then this just shows you a scheme on how we can integrate different predictions about drug response, and they can pick patients who are most likely to respond to the drugs that are being tested in the trial.
You said with the evolutionary genomics that there are that you might have a number of different types, subtypes of the cancer.
Yes.
Are we seeing high mutation rates after the initial mutations, or do we just have a number of different strains going on at the same time?
Unfortunately, one of the things that happens in cancer is often inactivation of DNA repair pathways or DNA chromosome stability and so forth mechanisms, and then you get lots of mutations, and those confuse. Those are called passenger mutations sometimes. They're just kind of along for the ride, whereas the original drivers that drove you to be a cancer were earlier on. And one of the big problems with cancer genomics is you see thousands and thousands of changes, but what were the important ones? But if you sequence, the passengers will be random and they'll vary from tumor to tumor. But now we sequence 1,000 breast cancer tumors with similar clinical features. The ones that appear again and again, those are the drivers.
So when the cancer genomics, David, have people been able to segregate population into better prognosis and worse prognosis based on the sequence? Is that starting to work?
It's starting to work. One of the big challenges with the Cancer Genome Atlas program, which is another parallel program, probably the largest government-funded, NCI-funded cancer genomics program, is now we're working on ovarian with them, and we're trying to draw out these Kaplan-Meier survival curves, and we're trying to predict survival better. It's very hard to predict survival from analysis of the tumor at this stage. So it's sobering.
But for some cases, like some of the leukemias, from RNA expression, you can actually classify tumors.
Well, that was the question I wanted to ask.
It's been really pretty amazing that you can separate them out that way.
Yeah.
I mean, RNA sequencing would be very effective at seeing this, because you would be seeing the deletions would come up as less RNA, the duplications come up more RNA, and things like that. And that's been working for quite some time, is to look at the RNA profile that predicts strongly patient outcome from a number of different cancers.
The data that we're working with now is mainly data from chips, and we're switching over to sequencing at this point. The mutations are only sequenced from selected cancer genes. We're not getting the whole genome very much, although we're going to get a genome a week next year. So it's picking up quite rapidly with the TCGA. And the future, the more or less consensus is that the future is a combination of genome sequencing and RNA sequencing. You want both. The problem with the RNA is that you're not seeing, if something's knocked out and you're not getting a transcript, you don't know that, but you would by sequencing the genome. And you get very different levels of expression, and it's very hard to decode everything that's going on in the genome from that. But the combination of the two will be extremely informative.
I have a quick question. Since you have a unique mRNA sequence in the cancer cells that you can identify as being unique to the cancer cells.
Yes, in case of a fusion gene, yes.
Would it not be an easy way to kill the cell by submitting a complementary RNA sequence that jams the production of that RNA and hence the following proteins and shuts down the entire metabolic process of the cell?
RNAi, yeah. RNAi is a therapy for other diseases as well, in particular, right? Complementary RNA. For large-scale use, delivery has been a problem, but if you're dying of cancer, you're willing to sit and have this pumped in, on a regular basis. So I think that's a great way to go for looking at these things. The idea of waiting before we get a small molecule that actually recognizes the fusion protein like we had to do in ABL-BCR is too long to wait. Okay, maybe Mike's company can make an antibody that recognizes the protein that's formed by the fusion transcript pretty fast, but the RNA would be even more direct. RNAi.
So are you optimistic then that the war on cancer will be won with this kind of technology? I mean, do you think there'll be a big impact on mortality from cancer from the future?
Yes. It's not going to be won in any one battle, but it will be the steady improvement. I hope I didn't suggest that all cancer is caused by these fusion genes, though. In a lot of cases, there isn't a fusion gene. There's a modified gene, a single base pair, or there can be just modification of the amount of expression of things can really screw you up. And the proteins themselves can be normal. So it's subtle, and it's a subtle interaction of complicated changes in pathways that we don't fully understand. That's why it's going to come slowly and not with one big bang. When? You're going to start to see, in a couple of years, you're going to start to see that, hey, we can treat this kind of cancer now. And yeah, sure, it's only 3% of the cases, but now we're doing a lot better. And then the next year, it's like, well, we got another couple of percent, show this particular genetic pattern, and we can actually deal with that this way. And so we'll just be picking off, and I think this process is going to be hot in certainly 5 to 10 years. There'll be just like every day, there'll be kind of a real phase of rapid progress where we're picking off subpopulations, certain understanding more and more different special types of tumors, getting specialized treatments for them. But this all depends a lot on the social things, right? Again, you can't go through the normal channels of, developing a small molecule drug and using it. There has to be different treatment options for this to really become fully mainstreamed. Yes?
Can you comment on what is the bottleneck? Is it a technical or knowledge-based bottleneck?
There is, actually it's primarily a knowledge-based bottleneck. We simply do not know how cells work. They are extremely complicated, and we cannot translate changes in the genetics, in the genome, to changes in behavior of the cell. We cannot make that leap at this point with any reliability in the vast majority of the cases. So it's a knowledge gap, I think. I mean, there's a technical gap now, but as we said, we're very confident we're going to overcome the technical gap. That will leave the knowledge gap.
David, some of your slides look like you had many cancers sequenced, and you had many breast cancers and many brain cancers. And then Mike put up a slide that said only 11 people have been sequenced.
Well,
I mean, are people sequencing more cancers than... Are scientists sequencing more cancers than people?
There will be probably next year more cancers sequenced than people. It's very possible. Unfortunately, every line, it looked like we sequenced the whole genome, but we actually that actually represented results from either a 1 million spot Illumina chip or the AFI 6.0, which you can get copy number information off of. So those were chip results.
That also goes to the question of what it defines a sequenced genome.
Yes.
I mean, there's a very, very fuzzy definition.
Yes.
So I know that one of the things that we're working on is establishing a metric that says, okay, 1 in 100,000 basically is a minimum basis.
Right. Yeah, very valuable.
The biggest difference I see between the RNAi method of combating these tumors and every other strategy of combating tumors, including traditional strategies, is the location. Instead of fighting it extracellularly, you're forced with the RNAi to fight it intracellularly, right? Mm-hmm. So you're worried about the delivery vehicle and how accurately you can target it. However, assuming that cancer hasn't reached metastasis yet, you only have to attack one or locate one cell and use it as a tag or use it as your ultimate localization vehicle.
Excellent. Yeah. Detection, early detection is probably the biggest gain.
As your zero-in mechanism.
Right. Yeah, absolutely. So actually, speaking of that, this is a segue into that. There are some interesting ways that people are trying to use the immune system to attack the tumor. So actually stimulating the immune system by getting it to recognize antigens that are produced by the tumor cells, and then your own body will attack the tumor in that case. So this approach is one of the exciting new kinds of applications of what we might think of as kind of a new immunology. And I think genomics is going to be a revolution in immunology. For, as we'll see, autoimmune diseases, organ transplants, and many other areas will be affected, perhaps much more than the cancer area. So we're not going to go through a complete description of the immune system here, but the key cells in your immune system derive from hemopoietic pluripotent cells that can either produce the white blood cells, the T and B cells, these T lymphocytes and B lymphocytes, or the natural killers, or it can produce some other key cells that are involved in early warning and involved in attacking and dealing with the invaders. And then it also produces just regular good old red blood cells. So one of the things that's interesting, if you look at the genomics of these cells, here's another case where something happens to alter the genomes of these cells in your body, right? So these immune cells, the T cells and B cells, won't look exactly like they did when they don't have your exact germline DNA. They go through a special process called VDJ recombination, and it's illustrated here. We're not going to go into the details of it, but you're born with your native genome with many copies, different versions of the V gene, different versions of the D gene, different versions of the J gene. And then what happens is when the immune cell is stimulated, it will go through this process of creating a variant of itself by cutting out a piece here, bringing a particular D in conjunction with a particular J that's shown in this box, and then it will cut out another piece here, and that will tack on a V. And now you have a triple, a V, a D, and a J, and that itself, attached to this little constant region that everybody gets over here, is now a very potent, now makes a very potent protein that can either in a B cell make an immunoglobulin that's going to recognize a foreign protein of some kind or foreign agent of some kind, or in a T cell, it will go into a T cell receptor or TCR that will also eventually serve the purpose of recognizing a foreign antigen. And yes, go ahead.
When do you think we'll be able to take an antigen that's on a surface of a cancer cell, say, and using computational methods determine the structure, like predict the protein structure that you would need to bind, and based on that predict the genetic sequence that you need, take a B cell, modify it, insert the correct gene, and then reinsert it, and so that the immune system doesn't even have to go through this process of guessing and searching.
That would be terrific. I think what we're going to see is, the protein structure prediction problem has been notoriously difficult for 30 years, and so my first guess is that we will develop technology avoiding having to solve that problem. But ultimately, I think a library of connections between common antigens, antigen being a small peptide sequence mostly, that stimulates the immune system. So that a kind of a mapping between antigens and particular VDJ recombinations that will be appropriate to build something that recognizes them is not out of the realm of possibility. I am actually a big proponent of trying to build such a giant library. One would have to do this from massive sequencing, so you've stepped ahead. I hope I didn't lose anybody here in this little interchange here, but just to go quickly through this, this is happening millions and millions of times in your body all the time. And because there are so many different combinations, we've only shown a few copies here, but there are dozens and dozens of copies, and so the ultimate combinatorial complexity of this is huge. Your immune system is capable of making trillions of different VDJ combinations. And so what happens is that when with all of these trillions, the one that actually responds to the antigen is selected and amplified by the immune system. It says, aha, well, this one matches, and then that cell proliferates, and now you have a robust immune response against this antigen. So it's a shotgun method. It just tries to make all kinds of different VDJs, but then they die unless they actually respond to an antigen, and then they get amplified. They'll actually go through more mutations and more amplification and so forth. I'm not going to go into the full details of this. The exciting thing, though, now is we can read what the immune system is doing. In other words, we can go in and read all these VDJs and say, this is what the immune system thinks it should be responding to now in this patient today. That will be revolutionary for immunology, I would predict. We are starting to look at some of this data again at Santa Cruz. This is data that we've downloaded from Robert Holt's lab in University of British Columbia. These are VDJs from T cell receptors, and you can just plotting possible J combinations with possible V combinations. Here you can see that this patient's blood is amplified for particular combinations, and then when you sequence this, you can actually look to see what that was, and you would translate this again into protein and try to figure out what that might be doing. I think we're going to need more of a lookup table rather than solving the protein prediction structure problem here, unfortunately. This area has enormous applications in my estimation. One of the most direct applications is patients that suffer from leukemia and lymphoma. This is where you have a cancer of the blood, where these immune cells are actually going cancerous, so you get a huge expansion of a particular type of immune system. You know that because they all have the same VDJ, and that is a immediate red flag that the person has a serious blood disease. If you suddenly lost your diversity and you're just making almost all the same VDJ, you are a very sick person at that point, and we will recognize that quite early. Organ transplant is a situation where the patient has to be immune suppressed to receive the foreign DNA, And it's very important to understand how they recover from that and whether they are mounting a response against the transplant. That holds for stem cell therapy as well, unless you're using perfectly patient-matched therapy. Whenever you get vaccinated, it would be very nice to know, did it mount the appropriate immune response? Have you lost that appropriate immune response, et cetera, et cetera? As many of you know, asthma, rheumatoid arthritis, and there's an enormous list of autoimmune diseases. These have actually become more and more of a problem in civilized kind of first world countries. They are all caused by inappropriate immune response, which could in principle be detected by full sequencing of the status of the immune system. And then I mentioned the therapy for cancer, where you're actually altering the immune system to attack what you want it to attack.